Good morning, everybody. Thank you for coming. Thank you for Sampson County Extension letting us using the building. Thanks to Dustin Kendall for helping spur this conversation and the idea generation for putting this event together. We only have 4 speakers this morning, so we should have plenty of time for discussion after each of the talks. We'll have a break as well to allow time for discussion. We'll go ahead and get started. I'm going to start out today, and I'm going to talk about the characterization of early life factors on sound lifetime productivity. Just to talk about a few projects quick going on at NC State that we're not going to talk in depth about today, but just to let you know that they're going on. In the upper left-hand corner there, we've done some gilt development projects in the nursery, looking at stocking density in relation to subsequent reproduction. Dr. Mark Estienne is going to be talking in depth about some other projects that they have done on the same topic later today. Heat stress, upper right-hand corner, we have a data set with prestige that we still have to finish analyzing. We've collected a lot of data looking at different factors related to heat stress. Sound body condition and management, we've done a lot of work there. With sound body condition, we're starting to look at some colostrum production work. We have that going on right now. And then Piglet quality, genetics of Piglet quality. Of course, we all know Piglet quality is important. We've been working with Smithfield Premium Genetics on a couple studies there. And genetic selection research lines. There's very few places in the United States that have the ability to have these genetic selection lines. NC State is one of them. Out at our Tidewater Research Station, we have one line right now we're selecting for reduced age at puberty. Of course, puberty is highly related to sound lifetime productivity. And I just checked the data this week, and we're on our 4th generation. And the control line through 190 days of age has 34% of the gilts that have come in heat. And the select line, 75% of the gilts have come in heat at 190 days of age, which is pretty exciting. So this is just some things that are coming down the pipeline or that we're working on. So if we look at the reproductive life of a female, we could break it up into the developmental period. As we see up above here, conception, that replacement gilt is born, then she's weaned, and then goes on to breeding, and then goes into the functional period or the breeding cycle. For this first talk this morning, I'm going to be focusing on the developmental period, and Dr. Harris's talk will also be focusing on the developmental period, focusing on this area from birth to weaning. So I'm going to be talking about 3 studies today, one that's published, 2 that aren't published yet, but we felt this information is important enough to get it out there and get it in front of you and help you guys get to start thinking about how you can utilize this information to improve sound lifetime productivity. So in gestation management, we're kind of working to get good birth weights, lactation management. The talk this morning, this talk is going to focus highly on pre-weaning growth. So if we can get those gilts to grow faster from the time they're born to the time they're weaned, when they grow up to be mature sows, they're going to produce more pigs and they're going to be more fertile. And then of course, anything we do to reduce age of puberty is going to enhance sow lifetime productivity as well. So here's the outline for my talk this morning, essentially linking pre-weaning growth and a few other factors to subsequent lifetime productivity. So back 40 years ago, Nelson and Robinson, they kind of came up with the idea, as we'll see here in the next coming slides, and then not a whole lot's happened. Not a whole lot happened for about 30, 35 years, and then we have 2 studies here just within the last decade, 2 other studies, both out of North Carolina, large-scale studies that we'll talk about this morning. So the Nelson and Robinson paper, they're the first ones to kind of look at the effects of that maternal environment. So looking at the effects of when that replacement gilt, when she's a piglet, what does the environmental effects on her as a piglet, how does that relate when she grows up and becomes a replacement female in the herd? This was conducted at the Tidewater Research Station out east in Plymouth, North Carolina, back in the 70s. So what Dr. Robinson did is he came up with strategic cross-fostering. So we'll say we have one sow that has 12, and we have another sow that has 8. He did some strategic cross-fostering to form 2 treatments. So in half the litters, he made them like litter size of 14, and the other half, he made them into a litter size of 6. So here's an example of how he would have strategically cross-fostered to produce half the litters with 14 piglets being nursed, and half the litters with 6 piglets being nursed. And just here, that's kind of what that would look like. Here's your treatment with 14 piglets. Here's your treatment with 6 piglets. And then took the gilts out of these litters and followed them up as they grew up and to look at their subsequent reproduction. And at weaning, this is 28 days of weaning here in 1970s. In the litters where there was only 6 pigs, those replacement gilts were 16 pounds at 28 days. And the litters where there are 14 pigs nursed, they're 13 pounds at 28 days. So about a 3-pound difference in that management strategy for that strategic cross-fostering. And so then he looked at the subsequent reproduction of those 2 treatments, and he found that the smaller litter sizes, gilts reared in those smaller litter sizes, had greater subsequent corpora lutea, number of embryos, and litter size. And to take a look at the litter size differences, so the graph is set up here, total number born alive. Here are your gilts that were raised in smaller litters, gilts that were raised in bigger litters. You see about a half pig to a pig difference. There's only about 60 sows in each one of these treatments, so not huge numbers, but for 1976, it's some good anecdotal evidence that there's something there. So not much was done for about 33 years, and then about 2005, Dr. Billy Flowers at NC State put together a study titled The Effect of Neonatal Litter Size and Early Puberty Stimulation on Cell Longevity and Reproductive Performance. So kind of taking OW's work, doing some of what he did, but then adding some layers on it too, and doing it in a much bigger fashion. This was done within an 80,000 sow production system here in North Carolina, so you can probably figure out where that's at. It was a 2x2 factorial, with one factor being neonatal litter size and the other factor age boar exposure, and this was funded by the National Pork Checkoff. So if you wonder where your pork checkoff dollars, this was some useful places that it went for this study here. So we again see our neonatal litter size was greater than or equal to 10 pigs nursed was one treatment. The other pig was less than or equal to 7 piglets nursed. So again, a large litter size or a small litter size, and then following those gilts out to see how they produced as they grew up and went on to become mothers. Age at boar exposure started at either 140 days of age or 170 days of age. So here's the results. The one bar here, we see the neonatal litter size of less than or equal to 7, so the gilts raised in small litters. Here's the gilts raised in large litters. This is weight at 22 days of age, and we see about a 4-pound difference at 22 days of age between those 2 treatments. And so in relation to reproduction, the gilts that were raised in those smaller litters, they went on to be younger at puberty. So within 28 days of boar exposure, 82% in the small litters came into estrus, and 60% in the larger litters came into estrus. And then they went on to have improved failing rate, about 5 percentage points, greater litter size, about a half pig, and superior sow longevity. So when you put all that together, that's quite a bit. Here is results from that study. This study was about 3,000 sows, so quite a bit bigger than the study from 1976. We can see here the effect of neonatal litter size on subsequent sow longevity. The blue line here is the gilts that were raised in small litters, and the red line is the gilts that were raised in large litters. And you can see the difference, where the big difference becomes apparent is right at R1 or rebreeding after that first period. And so then you can see the lines run somewhat parallel out through period 6, but big differences in longevity based on if that gilt grew up in a small litter or if she was reared in a large litter. And then here's the combination of effects. Effective neonatal litter size and early puberty stimulation on pigs produced through 6 parities. Here's small litter size, early boar exposure, over 40 pigs through 6 parities. And here's the large litter size and late boar exposure, about half as many pigs. So that the effects of the puberty stimulation and the litter size were additive. Here is some regression analysis that Dr. Flowers ran, the importance of pre-weaning growth characteristics on sow longevity. And you can see that pre-weaning gain, that gain from birth to weaning, and weaning weight explained an awful lot of variation in sow longevity. That's really quite, when you figure there's 100% of variation, and those two variables explain 35% of the variation, that's quite large. So now we're going to move into our third study, the effects of pre-weaning factors on sow lifetime productivity. This was conducted in a very large North Carolina system. It was a public university partnership, just as the Flowers study was. So it was an example of where NC State University individuals, researchers, were going out partnering with systems in North Carolina. This project also received from some pork checkoff funding, and then utilizing that pork checkoff funding to try and help find solutions and answers to improve our production systems here in North Carolina. So this study, so for the Robinson study and Flowers study, both of those studies utilized that unique cross-fostering strategy to create treatment differences. Well, this study didn't. It just kind of went out and observed what was going on. And so in the earlier studies of Robinson and Flowers, you have differences in the litter sizes that those gilts are reared in, but you also have a difference in the number of pigs that are nursed. So those two factors are kind of confounding. So what this study did of Cassidy's is you're able to sort out, okay, is it really the increase in pre-weaning growth that's causing the better reproductive lifetime, or is it the number of piglets nursed? So that's what this study helped to depict. So effects of pre-weaning factors on sow lifetime productivity, 12,934 gilts tagged. And traits that they looked at, traits of the dam of those replacement gilts, they looked at her parity, litter size, sex ratio of the litter, number of piglets nursed, number of piglets weaned, and then of that replacement gilt herself, looked at birth weight, whether she was cross-fostered, weaning weight, pre-weaning average daily gain, which are pretty similar, and then weaning age. And so here's a breakdown of the gilts in that project. 12,934 tagged, 82% of those were weaned, and then 48% of those entered the sow farm. So not all the gilts had an opportunity. They have extra gilts in that flow that they go to market if they don't need them, so that's why that's at 48% right there. And then of the 6,249 that entered the sow farm, 84% farrowed at P1, 63% farrowed at P2, and 43% farrowed at P4. And if we look through the North Carolina history of hog production, this happened in the last several years. So yes, these sows likely had some health challenges at some point in relation to PED and PERS. But we still found some significant results, which is pretty promising. So looking at retention from birth to parity 1, so including all 12,000 of those gilts, in the data analysis, improved retention was associated with a lower number of piglets born into the litter, which is an unfavorable association, but we're not going to be selecting for smaller litters anytime soon, so we probably don't need to worry a whole lot about that. Not cross-fostering, which I put an unfavorable sign because we like to cross-foster, but if we just cross-foster the males, well then they wouldn't impact the females. Improved piglet birth weight was associated with better retention from birth to P1. Of course, we want bigger birth weights, so that's potentially a favorable association. And then, of course, pre-weaning average daily gain was associated with increased retention to parity 1 as well. So if we don't count the gilts that didn't make it to the sow farm, we just look at the gilts that made it to the sow farm, how many of those reached farrow to litter. Then we see that again, total number born had an association, increased piglet birth weight, most of the same associations you saw on the last slide as you would expect. And then pre-weaning average daily gain explained the most variation of those gilts that entered the sow farm, of which ones that farrowed. This graph here across the bottom is piglet birth weight, and the side is retention. And the blue line here is the birth to parity 1, and you can see that line is quite steep. And then you see once they get into the sow farm, that relationship is a lot less steep, indicating that early on that birth to P1 is a lot of basically just piglet survival. So for the Cassidy study, if we look at throughput, sow farm entry to parity 4, increased piglets through parity 4 was associated with older weaning age. I didn't put a happy or a sad face here because you may or may not be able to change your weaning age depending on the system that you're in. The average age for the data set was 22 days, and so increase of 1 day in weaning age increased the number of piglets produced through 4 parities by 0.32. So there's been some work done at NC State that shows around day 24 that piglet has a better developed gut, and so that somewhat ties into the recommendations here we make here in a few minutes. And then of course pre-weaning average daily gain. Increase in pre-weaning average daily gain of 1 pound at weaning was associated with increase of 0.38 piglets through 4 parities. So in summary, from the Cassidy study, cross-fostering gilts, if they were cross-fostered, they were 2.45% less likely to farrow litter. Increased birth weight, increased gilt retention, increased weaning age, increased reproductive throughput, and increasing pre-weaning average daily gain, increased reproductive throughput, and that explained the most variation of any of the factors. So implications on multiplication farms, if you were to look at the data from this study, you would say not to cross-foster the male, just only cross-foster males and not cross-foster females, because it potentially reduces their reproductive lifetime. And then improve piglet survival to increase the size of your gilt pool. Increase weaning age if you're less than 24 days, according to this study. And then, of course, improve pre-weaning average daily gain. And if we look at all 3 studies, the Nelson and Robinson, Flowers and Cassidy, all 3 show that increased pre-weaning average daily gain improved subsequent lifetime reproduction. So now, obviously the question is, so how are we going to go about this? How to best do that? Is that going to be done through strategic cross-fostering? We're going to some gestation lactation management, increase colostrum production, modify our lactation feeding somehow to improve pre-weaning average daily gain. Dr. Harris is going to talk about supplemental feeding of piglets. What's the best strategy? Is there economical strategy to do this? So the strategic cross-fostering actually is being conducted in at least one system that we know of within the United States. It's an internal multiplication system where they're able to take barrows off those replacement gilt litters and put them onto commercial litters. So with that, I thank you guys for your time and be happy to open any questions or discussion that you all may have for this first presentation.
Cross-fostering the barrels only, is that just to help the sow, try to get them more milk and gills that are left?
So the question is, cross-fostering barrows only, is that to help the sow? Well, piglets a lot of times link up with a teat, and so if you cross-foster the gilts, there could be some stress, or maybe it takes them a bit to establish a teat. Dr. Flowers, you want to add anything onto that?
So, I think it does both things. It helps the sow, and her milk's better milk, more milk than the gilts are less. That helps the breeding growth. So, in most systems, we could argue this, but a maternal line barrow really has less value.
What do you do with the sow that was nursing 6 gilts in the subsequent farrowing?
The question is, what do you do with the extra pigs?
No.
No.
With the sow that wasn't challenged enough because she was nursing only 6 gilts, what happened with that sow in the subsequent farrowing when she has to nurse 14?
Oh, okay. So the question is, if she nurses fewer in one parity, what happens in the subsequent parity?
Yeah.
There hasn't been a ton of work on that.
In our study, there's no difference. So there's this perception that you can load up sows early in their life that's improve their subsequent reproductive performance, and it really hasn't been very good controlled studies to substantiate that. So, I mean, the memory rate is supply and demand. So it's either on or it's off.
So Sung-Woo Kim has done one study, was a smaller study. There's one other study published, but yours would be by far the biggest data set. And so that does, I would agree that there seems to be a little bit of a myth that you have to load up sows. And actually, we just ran a huge data set, and if you load up P1 sows, it obviously hurts their return. And that's what this one system's doing that we need to talk with to get it corrected. But good question.
So for the commercial farms that this wouldn't necessarily affect us, our take-home message would be to possibly expose those gilts to boars earlier in their development, like in the gilt developer stage.
Correct, but definitely on the multiplication, what's going on there is going to impact you guys.
Right.
Okay, I got a question. So if you got a 14 live one and they're all loaded up with 14, how you gonna manipulate that? You gonna lose a bunch of pigs? You got a farm with 13, 14 live one and they're loaded up, even at multiplication. You're stuck. That's the question.
Yeah. No, that's definitely the question. No, you're definitely right. And that's why we brought in Dr. Harris. Dr. Harris to talk about potential artificial rearing strategies, what's...
Yeah, and there's some milk products that will work on that if you want to try that.
What's the answer here?
Yeah.
So,
I mean, because multiplication's got the highest mortality in the animals we got in our system. Because their genetic area's quicker, their live feeding. We got people run out of ears. And we're not going to operate it. So we're kind of stuck.
with that, we'll move on to our other speaker. We got an hour for her, so we'll probably definitely have time for more questions there. So, Dr. Harris.